Okay, welcome back, students. This is Dr. Balding. One more section to this genetics chapter. Now we're going to talk about some types of genetic alterations that we see, specifically genetic transfer. And so, what we're talking about here, you guys remember in the beginning, I talked about how bacteria especially were notorious for being able to transfer genetic material laterally, okay, or horizontally. So, of course, daughter cells inherit genetic material from their mother cells or parent cell. So there's vertical transmission of genetics or genetic material. Right, but there can also be a lot of lateral transfer going on in prokaryotes. So we're going to talk about mechanisms in which you can do lateral gene transfer. And so these mechanisms include a process known as transformation, a process known as transduction, and another process that we call conjugation. Okay, and so these are the three processes of lateral gene transfer that we're going to address. Okay, so the first one is transformation. Transformation comes from an interesting story of a guy back in the 1920s. His name was Avery Griffith. So Griffith was studying, and it was at that time a booming age of vaccines. There were all kinds of vaccines in development. So for every pathogen that was discovered, scientists were aggressively looking for vaccines to prevent that disease. And so Griffith was looking at an organism called Strep pneumoniae, which can cause pneumonia, as its name implies. And he had a mouse model of infection of pneumonia. So if he injected mice with live virulent pneumococci, then he could show that it would kill the mouse. Okay, so in vaccine development at the time, and still today, sometimes we take organisms and we will kill them and inject them into our model organism and see if, upon challenge with the live organism, are they able to survive. And so he had a model where he could heat kill these pneumococci and demonstrate that upon challenge the mouse would live. And then he found another candidate for a vaccine, and that was, he noticed that some of the colonies of bacteria on plates looked what we call rough. Okay, they were dull, they weren't glossy like the smooth pneumococci, the normal colonies, and that's because they didn't have a capsule. Capsule actually gives colonies or the smooth, glossy, mucoid appearance. Okay, so they didn't have capsule, one of the virulence factor they need to evade their host. So he injected those into the mouse, challenged with a live bacteria, and then the mouse lived. So he had two vaccine candidates here. Well, Griffith did an experiment, a control experiment well, actually, maybe it wasn't a control. It was an experiment to see if he could increase the efficacy of the vaccine by mixing these two vaccine candidates together. And so what happened when he mixed the heat-killed bacteria with the live but non-capsulated bacteria, what he observed was his mouse, upon challenge, actually his mouse didn't make it to challenge, his mice died. And he was able to recover bacteria that didn't look like either one of these. They were live okay, and they had capsule. And so Griffith proposed that they were somehow transformed. He suggested that the heat-killed smooth pneumococci didn't come back to life, okay, that he had killed them and they were definitely dead. But he suggested that somehow these heat-killed smooth bacteria were able to donate some genetic information to these guys that were living that didn't have a capsule to allow them to produce capsule. And he called it the transforming factor. Well, it wasn't until the 1950s, or maybe it was the 1940s, about 20 or 30 years later on, Avery, McCarty, and McLeod were able to demonstrate that this transforming material was DNA. They basically took the cell soup, digested it with either DNases, RNases, proteases, and other biomolecule degrading compounds, and shown that only when DNase was added were they able to eliminate the transforming material, okay, which turns out to be the genetic material. Okay, so there are bacteria that take up DNA really well from the environment, and they're called naturally competent, including strep, which is why Griffith's experiment worked so well, and also bacillus, and an organism that I used to work on, Neisseria meningitidis and Neisseria gonorrhea. Those guys take up DNA from their environment pretty well, and you may say, well, why would they do that? Well, to some extent, it's an advantage. It can be detrimental for them to take up foreign DNA and incorporate it, because they could harm their genome. But if you've got a huge population of these organisms, and say the immune system attacks, if one has some variation, it is in the interest of the entire group that they vary a little bit. So maybe this particular one that has DNA from another organism can survive, and sometimes they can pick up, toxins from other organisms, and we'll talk about that later on. And sometimes they can pick up antibiotic resistance mechanisms and so forth. So being naturally competent can be a benefit. And you may think, well, is there normally a whole bunch of DNA just laying around for cells to pick up in their environment? And actually the answer is yes, there is. In biofilms, there's a variety of different organisms, and they're oftentimes in very different stages of life. Some may be dying, some may be growing and dividing. And so, yeah, there's always dead bacteria around for other bacteria to, acquire their DNA. Are they all competent? No, they are not. Even E. coli that we work with in the laboratory is not a competent organism. It actually can be made competent, though, to take up foreign DNA. It can be artificially induced. And the way we do that in the lab is that we kind of treat the E. coli badly. We will take it and grow it up in a nice rich media to get the cells to grow, but then we subject it to a hypoosmotic condition like water or low salt solution, and make them very, very cold and spin them over and over again in this cold environment, and then add the DNA. And that stress causes them to take up foreign DNA. And they can take up genomic DNA, plasmids, and this is how we can transfer plasmids from strain to strain very easily in a science lab. Okay? But remember, this is a way in nature that they can do this as well. So we're going to talk about another mechanism of genetic transfer that we call transduction. Now, transduction is going to involve the use of bacteriophage. So this is the normal life cycle of a phage, where this phage will attack this host bacterial cell. It will inject its DNA into the cell. Notice that the phage head, the empty phage head or ghost, remains on the outside and gets degraded in the environment. But when this viral DNA is injected, it basically takes over the cell. It'll cut up the host chromosome into little pieces and then make viral copies of its own genome that get packaged into viral particles, as you can see here. Well, most of these viruses have the green color DNA, which is viral DNA, but occasionally, accidentally, this phage packaged chromosomal DNA, which came from the E. coli host, right, or salmonella host, or whatever bacterium it was. Now, when these phage burst the cell open, right, so it pops open from in the normal life cycle of the phage, and these are released, these normal phage are going to go on to do the same thing over in a new host cell. But this guy who got chromosomal DNA doesn't have the viral genes to take over the cell. Instead, what happens is he injects E. coli chromosomal DNA into the host, which can be integrated into this host cell, and now this cell has received some DNA from a different host and may be able to utilize it to its advantage. Okay, it's probably done in some sort of allelic exchange and has new genes that it didn't before, or it gets has an insertion and an addition of DNA that it may not have had. So this happens on occasion as well. These are all rare events, but happens enough that it can be problematic, especially in medicine. Okay, so another type of genetic transfer, or lateral transfer, is caused by conjugation. Now, conjugation, as we may recall, I've talked about conjugation pili, or sex pili, that are formed are going to involve fertility plasmids and sex pili and so forth that I talked about before, but we'll just review this a little bit. So here we have an F plus cell that has an F plasmid in it, an F minus cell that is fertility minus, so it doesn't have that plasmid. Genes on the plasmid encode for the formation of this sex pilus, which is very short here, but that's that little gap that you see there. There's an enzyme encoded by this plasmid that nicks the DNA and allows a single strand to pass through that sex pilus into the F minus cell, right? And so as this single-stranded DNA comes in, it's repaired by the host. The single strand left behind is repaired by that host, and now we have two fertility plus cells. Well, like I said, on a rare occasion, these F plasmids get integrated into their host chromosome, as we see here, and become part of it. And when they do, they're no longer F plasmids, but they're called HFR, or high frequency of recombination strains. And when conjugation occurs and HFR meets up with an F minus cell, the genes are still there to form the sex pilus, right? But instead of the plasmid getting nicked and transferred, then we have at the origin of transfer here, the host chromosome gets nicked and transferred into the recipient F minus cell. And so this is what we see going on in this diagram, where you have the nicking and single strand transfer. And so you'll notice that F minus cells have alleles A and B down here, right? But so does the high frequency of transfer strain, okay? And it's close to the ORI. And so these A and B genes get transferred and recombined with the A and B alleles of their recipient host, and now A and B are incorporated into this F1, we call it recombinant bacterial strain. So these may be the same alleles that this organism had before, or they could be different alleles of that gene and give this organism a different genotype and phenotype. Okay, and so that's lateral gene transfer. by 3 different mechanisms. Now, there are also events that occur in the cell, major DNA movement events that we call transposition, which can cause major genetic changes in a cell. There are these individual pieces of DNA called insertion sequences, or IS elements. Larger ones are called transposons. They're a little bit more complex, that can mobilize DNA from one place in a cell to another. It was originally discovered in corn, and that's why the corn kernels are all shown down here. You've probably seen this corn at Thanksgiving time in stores and so forth. You have a darkly pigmented corn kernel that has anthocyanin in it. There are other corn that you see here that are white that don't make anthocyanin, and some that are sort of intermediate. Well, the gene that's responsible for making this color pigment gets interrupted by one of these insertion sequences. And so it doesn't make the color, or it may just be one allele gets an insertion and not both of them. And then you have sort of an intermediate color. But these were originally discovered by a lady named Barbara McClintock. She received the Nobel Prize in 1990 after she discovered these things back in the 1950s. Her work was rejected for a long time because it wasn't believed that DNA could do this, that it could be mobile and move around. DNA was known to obtain mutations, but not major genetic mobilization like this. So that was a unique idea. So let me show you what IS elements do. IS elements like this one here, IS1, which was actually the first one discovered in bacteria, they're numbered according to the order in which they're discovered, can mobilize itself. So it can copy itself and actually move into a new target sequence. So you see this IS1 element right here on the DNA, and this is just showing you folded up. And it's folded around a protein called a transposase, right, that catalyzes its movement. Right, and so what happens is this transposase will cleave the ends of the target DNA and copy the IS element and allow its insertion into this new target site. Right, so it has increased in copy number. Some of them just move without copying, so they just move from one location to another, and others copy themselves as they grow and increase in number in the cell. So there are IS elements. Transposons usually not only carry the transposase gene that encodes for this protein and just the ends, but transposons also oftentimes carry antibiotic resistance markers, so they mobilize them. Like I said, these can cause major genetic rearrangements. A complex transposon looks like this, where you have an IS element and another one here, IS1. And what happens is they could be actually far distant on a piece of DNA, but when its transposase is expressed and gets ready to cut this IS element out of the genome, it can cut here and here to mobilize it, or right, it can cut here, like here, and over here, because this is the same element, right? It has the same ends. And so instead of mobilizing just this piece, all right, in this instance, when you cut here and here, you mobilize the whole thing. And notice that IS element has something important in between. It has a kanamycin resistance gene. So it doesn't have to be just one gene. There could be hundreds of genes in between these two IS elements that can be mobilized to another chromosome. They can be mobilized onto an F plasmid that can then be mobilized into a new host. Okay, so it's a way in which genetic rearrangements can occur And this actually occurs much more frequently than any of the other processes that we talked about, the transduction, transposition, or transduction, transformation, or conjugation. All right, so that ends my discussion of genetics, topics that we're interested in for medical reasons. So if you have any questions, please let me know, guys.